In this video, we will discuss about beta oxidation of fatty acids. We can also write beta like this. Now, fatty acids, they are part of lipids or fats. Now, they are of two types, saturated and unsaturated. Saturated when they do not have any double bonds, like if we write any fatty acid, if we open the carbon chain like this, now here there is no double bond, so this is saturated fatty acid. And if it is unsaturated, this can be like this. This can have double bond, it can be 1, 2 or 3 double bond, then it is called as unsaturated fatty acid. Now in this video we will take the example of saturated fatty acid. Now saturated fatty acid which do not have any double bond, depending upon their carbon length, they are categorized into 3 categories. Short medium chain fatty acids whose length varies from 2 to 12 carbon in length. Long chain fatty acid, number varies from 14 carbon to 20 carbon in length. And very long chain fatty acid which is having more than 20 carbon length, so they are called as very long chain fatty acids. Now we'll discuss certain examples, like certain examples of fatty acid which are varying from 2 to 12 carbon length fatty acids. If the carbon number is 2, this is called as fumaric acid. If it is 3, it is propionic acid. And if it is 4, it is butyric acid. And if it is 6, this is caproic acid. If it is 8, caprylic acid. 10, capric acid. And 12, lauric acid. And if the number varies from 14 to 20, this can be myristic acid, 14 carbon in length, palmitic acid, 16 carbon in length, stearic acid, 18 carbon in length, and arachidic acid, 20 carbon in length. So these are different kind of fatty acids. Now what is fatty acid oxidation, right? Now these fatty acids, they are broken down into units of acetyl coenzyme A. So acetyl coenzyme A, CH3CO, this Kugel bond, S coenzyme A. So this is having 2 carbon compound length, so it is called as 2 carbon in length. Acetyl coenzyme A, which is released by oxidation of fatty acid. Now this can either enter into Krebs cycle or it can form ketones, and this can be also utilized for the metabolic fuel. Now we'll discuss in detail what is beta oxidation, right? If we take any of the fatty acid, it can be any fatty acid like R, CH2, 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 COH. So here we can take any length. It can be 14 carbon, it can be 16 carbon, it can be 18 carbon, 20 carbon, 12 carbon, so on, right? So we can take any fatty acid. Now this is the acidic group, and adjacent to this carbon, this is called as alpha, and this is called as beta. So at this position oxidation is going to take place, so this is called as beta oxidation. So we can take the example of palmitic acid. So we can see here the 16 carbon in length. So this is 1, 2, 3, 4, then 11 carbon, 15, and this is the 16 carbon. So this is palmitic acid. We can take any of the fatty acid. Now where does it occurs? Now this fatty acid oxidation occurs in the cell organelles like peroxisomes, in plants and fungi, and in seeds it is in glyoxysomes. We can see here this is the peroxisome, this is a membrane bounded cell organelle, this is crystalline core, and this is glyoxysome. Again this is membrane bounded cell organelle. So in seeds it is in glyoxysomes, in plants and fungi it is in peroxisomes, but in mammals and animals mostly this is into the mitochondria. Now mitochondria, they are also called as powerhouse of cell. Now short chain fatty acid can directly enter into the mitochondria, but medium chain and short chain they can enter, but long chain fatty acid they cannot enter, right? So they need special we can say carrier for their entry into the mitochondrial membrane. So this is called as carnitine shuttle, which helps in entry of fatty acid into the mitochondria. So this is the cell organelle called as mitochondria, which is bounded on outer side by this is outer membrane, and this which is being folded like this is inner membrane, right? So here in the matrix of mitochondria, fatty acid oxidation will take place. First, these any of the fatty acid which has to enter into the mitochondria, that is activated. Like this is any fatty acid. Now acyl coenzyme A synthetase enzyme will activate this fatty acid, and here at this part squiggle bond and coenzyme A is added. So this is called as fatty acyl coenzyme A, and for this energy is required in the form of ATP because coenzyme is being added. So this is here being added, and magnesium and chloride ion are also required for its activity. So fatty acyl coenzyme A become activated. Now this can enter into the mitochondria, and here fatty acid oxidation will take place in case of animals or mammals. Now we'll discuss the mechanism of beta oxidation. How does it occurs? It has actually 4 reactions: oxidation, hydration, dehydrogenation, and thiolysis. Now we'll discuss in detail. Now this is the fatty acid, like we discussed, any of the fatty acid. It can be of any length, 14 carbon, 16 carbon, 18 carbon, right? It can be any of the length. So this fatty acid is activated by acyl coenzyme A synthetase, and this gets converted to fatty acyl coenzyme A, and it enters into the mitochondria where its oxidation takes place, or in any cell organelle where it is going to be oxidized. Now we can see here, this is the alpha position and this is the beta position, right? At this position, between the two, alpha and beta, double bond is being created. And how this double bond is being created, I have shown here. We can see here hydrogen and hydrogen. They are being removed by desaturase enzyme, right? And this part is being converted into double bond like this here. So this is also called as oxidation, or also called as dehydrogenation. I mean, 2 hydrogens, they are being removed, and enzyme which is catalyzing this reaction is fatty acyl coenzyme A oxidase. And these 2 hydrogen which are being removed from here, now they will be accepted by flavin adenine dinucleotide, and this will get converted to FADH2. So this is flavin adenine dinucleotide, and this is being reduced accepting the 2 hydrogens, right? So here double bond is being created. So this is called as trans delta 2 enoyl coenzyme A. I mean, here at this position double bond is there, beta, and within beta and alpha. Next step is hydration, right? I mean, water is being added. So enzyme required is enoyl coenzyme A hydratase. Along this double bond, right? Here OH is being added and here H is being added. I've shown here how this is happening. So this will change into beta hydroxy fatty acyl coenzyme A. Now this is coenzyme A, this is CH2, here H is added and here OH is added, right? So this is called as beta hydroxy. I mean, at beta position hydroxyl group is there, right? So this is called as beta hydroxy fatty acyl coenzyme A. Now here, this is OH and this is H, right? Again, 2 hydrogens will be removed and keto group will be created over here, right? So next step is dehydrogenation again. I mean, hydrogens are being removed at this position, right? This and this. And NAD will accept these 2 hydrogen and get converted to NADH2. And enzyme required is beta hydroxy fatty acyl coenzyme A dehydrogenase, right? And this will get converted to beta keto fatty acyl coenzyme A. We can compare these 2 molecules. This is same, right? Here it is same, but at this position keto group is being created like this, right? So this is called as beta keto fatty acyl coenzyme A, right? Here I have shown how this keto group is being created. Now fatty acyl coenzyme A acetyl transferase. This is also called as thiolysis or acetyl transfer reaction. I mean, acetyl is being transferred. Now we can see here this part of the molecule. I'll just make like this. This part of the molecule, right? Now this will be removed. CH3COS coenzyme A. This will be removed from here. I'll again make like this, right? This will be removed. And we can see here, this part is being removed, right? This part, like we have shown here also. So this part is here like this. We can see here. So this acetyl coenzyme A has been removed. So when this acetyl coenzyme A have been removed, right? So what part of the enzyme, what part of the this molecule will be left? Up to this molecule. This will be left, and this has been removed after that, right? So coenzyme A is being added, and coenzyme A will be added over here at this position. And this 2 carbon has been lost in the form of acetyl coenzyme A. If we started from 16 carbon, now this will be 14 carbon in length. If we started from 18 carbon, now this will be 16 carbon in length, because 2 carbon length have been removed in the form of acetyl coenzyme A, right? So this is how fatty acyl coenzyme is again regenerated. Now this has become 2 carbon less in length. So now again this will be the alpha position and this will be the beta position. And the whole cycle will be repeated like we have done all the reactions. So after 14 carbon और जो भी लेंथ है उसका जहाँ से भी हमने स्टार्ट किया, चाहे हमने 12 से स्टार्ट किया, चाहे हमने 14 से स्टार्ट किया, 16 से स्टार्ट किया, 2 लेस होता जाएगा, 10, 12 and 14, right? So ultimately we'll get fatty acyl coenzyme A, right? And here NADH2 is being produced, and here enzyme involved is fatty acyl coenzyme A acetyl transferase, and this is the acetyl coenzyme A which is being removed. This can enter into the Krebs cycle or for ketone formation. So this is about beta oxidation. Again, I'll quickly revise. This is any fatty acid of any length. First, it is oxidized or dehydrogenated, right, by the enzyme acetyl coenzyme A synthetase. Now this will be converted to trans delta to enoyl coenzyme A. Now hydration will take place, water is added. This is beta hydroxy fatty acyl coenzyme A. Again dehydrogenation will take place. This will get converted to beta keto fatty acyl coenzyme A. Acetyl coenzyme A will be removed. This is called as acetyl transfer or thiolysis. And again fatty acyl coenzyme A will be produced. So 4 reaction has occurred: oxidation, hydration, dehydrogenation and thiolysis, and acetyl coenzyme A have been removed. If we started with, supposing we started with 16 carbon, 2 carbon have been removed, so this will become 14 carbon. In the next step, again 2 carbon will be removed, 12 carbon, right? So we can divide by 2, right? 6 times this will occur, and 2 we can say CH3COS coenzyme A of 16 carbon. How many acetyl coenzyme will be produced? 8. Right? So 8 acetyl coenzyme A will be produced of the 16 carbon. If it is 18 carbon, this will be 9. If it is 20 carbon, this will be 10, right? So whole of the fatty acid will be converted into acetyl coenzyme A. Now which can, again coenzyme A can be removed, this can be acetyl. Now this can enter into the Krebs cycle or any other metabolic processes. So overall reaction will be, now we can discuss here. This is palmitoyl coenzyme A, this is 16 carbon in length. We need 7 coenzyme A, 7 flavinadenine dinucleotide, 7 NAD molecule nicotine adenine dinucleotide, 7 molecules of water, and we'll get 8 acetyl coenzyme A, 7 FADH2 and 7 NADH2. And if we are taking sterile coenzyme A, which is 18 carbon in length, we will need 8 coenzyme A, 8 FAD, 8 NAD, 8 molecules of water, and 9 acetyl coenzyme A will be generated, 8 FADH2, 8 NADH2. So ultimately, fatty acid, जितनी भी उसकी लेंथ होगी, ultimately that will be converted into acetyl coenzyme A. अगर 20 है तो 10 बन जाएंगे, अगर 18 है तो 9 बन जाएंगे. So whole of the fatty acid will be oxidized because this is occurring at beta position, so this is called as beta oxidation. I hope everything is clear to you. If you have any question, please ask me in the comment box, and thank you for